Hi everybody. So we're going to continue with your lecture on the urinary system. To just quickly review what we discussed last time, we talked about the fact that urinary system is, your kidney has three main functions, and those three functions are glomerular filtration, tubular reabsorption, and tubular secretion. All of these three steps leads to basically production of urine in your body, and that urine eventually be excreted through the ureter, urinary bladder, and urethra. We talked about the glomerular filtration, and one of the facts that we discussed was that this is a passive process and allows only the molecules that are small enough to pass through the capillary within the glomerulus and basically get into the capsular space and eventually enter your proximal convoluted tubules. Um, Something to note again is if you look at blood component contained plasma, red blood cells, white blood cells, and platelets. And we mentioned that red blood cells, white blood cells, and platelets were too large to pass through and become part of the filtrate, while all the components of the plasma were small enough to do pass through to become part of the filtrate by passing through the capillary network, with the exception of proteins. Proteins will be would remain inside the blood and will not become part of the filtrate. And again, note that you will test positive for red blood cells, white blood cells, and proteins in your urine if there is damage to the glomerulus, which causes these really large components to actually pass through and become part of the filtrate and eventually part of the urine and excreted from the body. Now, we also talked about the three main pressures associated with the urinary system. We said two of these, more specifically, it's your nephron to allow the filtration to happen. The largest pressure is associated with the afferent arterial pressure connected to your aorta, and this is hydrostatic pressure, which ranges about 55 millimeter of mercury, pushing fluid out into the capsular space. And while this pressure is pushing out, there's two pressure working against, uh, basically pushing fluid back into the glomerulus. The two pressure are, the larger one is the osmotic pressure, which is basically you have a lot of solute molecules inside your blood, so water is going to be pulled back. This is a pressure of about 30 millimeter of mercury. And the smaller one is basically the pressure of water within the capsular space that is being exerted onto the glomerulus. This is a smaller pressure, about 15 millimeter of mercury, because the amount of fluid that is present in the capsular space is a small, and therefore the hydrostatic pressure will also be small. So the net filtration pressure is about 10 millimeter of mercury, basically 55, subtract 30 and 15 from it, and that will give you an average of 10 millimeter of mercury. These are the components that will be visible in your tubular filtration. Notice that basically anything that is in the plasma is part of the filtrate, with the exception of the proteins that are too large to be part of the filtrate. Now we're going to take a look at the second process associated with the urinary system on formation of urine, which is a process called tubular reabsorption. So let's start by looking at the word reabsorption. Reabsorption implies returning or reabsorbing water and solutes from the renal tubules, basically your PCT, your descending and ascending limb, and your ECT, back into your circulation, which is your bloodstream. And this process of reabsorption takes place in the peritubular capillary network. To remind you, the peritubular capillary network, which is also the same thing as vasa recta in the juxtamedullary nephron, there are the tubes that are found around renal tubules. These are extremely important for the process of reabsorption as well as secretion, as we learned later on. So here is a picture that describes the two membranes associated with the process of tubular secretion. So what you see right here, these are the cells of the urinary system. So let's say this is representing your PCT. So this is basically the cells that are lining your PCT. Where it says lumen, this is where your filtrate can be found. And where it says extracellular fluid, that's basically the space. And logically, this will become also your bloodstream, where you basically transport molecules from. So the way I want you guys to look at it is this. You have filtrate, which contains water, ions, glucose, and amino acid right here. So you need to take those molecules and return them back somehow back to the bloodstream that you have right here. Now, to understand this, we have two membranes, and these membranes are very important because of the type of protein channels that can be found in them. Now, on this side, which is facing the lumen, these are basically the surface or the membrane that is referred to as the apical membrane, which we learned about it earlier. And at the base of the cell and on the sides of it, we have a membrane referred to as basolateral. Baso, base part, lateral on the side. So basolateral, base and lateral side of the cell. Do notice that these cells are held together with a tight junction. And if you guys remember from earlier on, tight junctions are very important for preventing molecules to basically pass through. So you're limiting molecule movement. Although do note that there's always exception to the rule, and there are molecules that have the ability to permeate the tight junction and cross to the extracellular fluid. So let's continue by looking at the path that the water and solutes can take to again go from the lumen, which is where your filtrate is, into your bloodstream, which is ECF. So we can take two paths. One of them is transcellular route. Transcellular implies that you're going to go through the cell. So it's through the cell. So right here, transcellular is basically you're passing through the cell. So if I have, and I have a picture and I'll show you just a second, but let's say this is your cell. This is the apical size. This is the basolateral, which means this is your blood and this is your filtrate, okay? So if the fluid and the solutes take this route, okay, this is transcellular, which means you have filtrate, it passes through the apical membrane, it passes through the cytosol of the cell, it comes out through the basolateral membrane, it passes through the interstitial fluid, which is right here, and get into your blood capillaries. So interstitial fluid is this gap, which is again part of the ECF. So if you guys, again, I'm just gonna recall this. ECF, which is extracellular fluid, is made up of plasma, which is part of the blood, and interstitial fluid. If you guys don't remember this, please go back and review your lectures when we talked about cell structure. This was one of the slides that described this concept in a lot detail. Now the second option we have is paracellular route. Paracellular route, we're going to draw another cell next to it. So paracellular is basically if you have tight junction right here, tight junction right here, what it's going to do is allow the molecules to pass through the tight junction and get into the bloodstream. So you're not going through the cell, but you're going between the cell by passing the tight junction. And typically waters and ions are the ones that are allowed your tight junctions is permeable to, and basically allow these molecules to move from the filtrate into your bloodstream. Here's the same picture I've just described to you guys, so just kind of reviewing it one more time. You have filtrate right here. Filtrate. I guess I should just point out to this. Filtrate. You have the apical membrane. You have the basolateral membrane. You have the interstitial fluid right here. And you have your blood capillaries right here. So water and solutes are entering the filter, passing through the apical membrane, through the cytoplasm or cytosol, out of the basolateral, into the interstitial fluid, and eventually into your bloodstream. Versus the second route, which is the paracellular route, where you're passing through your tight junctions. So notice you have like tight junctions, same thing right here. So you're bypassing or leaking through your tight junction to eventually end up in your bloodstream. Now, as far as reabsorption, we actually can use two techniques to move these molecules in terms of energy. Not in terms of the route they take, but rather the energy they consume to be moved from the filtrate into your bloodstream. One, if you guys remember, is a process called the active transport, which in this case we refer to it as active tubular reabsorption. If you can recall, the word active implies we need some sort of energy, so you could use the energy in a form of ATP, or you can use a secondary, which basically you moved with one ion with its concentration gradient while the other one moved against concentration gradient. And secondary was branched into, again, it was referred to as either antiporter or symporter. So symporter, basically both molecules moved in the same direction. One with concentration, one against concentration gradient. And antiporter was the molecules moving opposite of each other. Now, as the word passive implies, passive tubular reabsorption is movement of molecule without the need of energy. And the best way or the best example of that would be diffusion, osmosis, and facilitated diffusion. Diffusion was basically movement from high concentration to low concentration without the need of membrane proteins. Osmosis is diffusion of what? Sorry for the writing. Diffusion of H2O. And facilitated diffusion is the same thing as diffusion, but with the help of what? Help of proteins. So this is very important to remember as far as the difference. Apparently I can't spell, guys, sorry. So let's take a look at reabsorption of the molecules, and then what we're going to do is we're going to look at it at each segment and see what gets reabsorbed at what section, at what percentage. So tubular reabsorption of sodium, it is, do remember that sodium is one of the most essential ions in your body. It is the most abundant one in the filtrate, and about 80% of the ATP that's going to be used in the kidneys, or more specifically in the case of nephron, is to allow for the sodium reabsorption. If you guys remember the sodium potassium ATPase, right? 3 sodium goes out while you move 2 potassium in. This is a mechanism that basically again uses transcellular transport. Sodium is reabsorbed through the renal tubules, and I'll explain this in more detail, from basically the filtrate, and it gets eventually reabsorbed into your, it returns back into your bloodstream. And depending on the segment of the renal tubules, the sodium reabsorption is significantly different. And do keep that in mind. It's extremely important to remember this, because depending on the region, you either have really high levels of sodium reabsorption or no sodium reabsorption, period.